Okay, so this is a skeletal muscle cell. Let's go through and identify as many structures as we can. First off, this right here, this is the axon of a motor neuron. Now, in order for a skeletal muscle cell to contract, we need to have some sort of nervous input. That's going to come from the form of a motor neuron. So here's the axon of a motor neuron. Now, when we get to the nervous system in the next section, you'll learn to identify that the cell that surrounds the axon of a motor neuron, these are called myelin sheath cells. But for our purposes right now, this is the axon of the motor neuron. It wraps around. This is still axon of the motor neuron. And then it comes and branches into multiple axon terminals. And it's the axon terminal, this structure right here, the axon terminal which synapses with the sarcolemma, which would be in white here. So axon terminal, sarcolemma, which is the plasma membrane of the skeletal muscle cell. So these axon terminals come down and form a synapse with the motor end plate of a skeletal muscle cell. Now, I know that this is the axon terminal of the motor neuron because of the presence of these synaptic vesicles. These little yellow dots represent vesicles, called synaptic vesicles, filled with the neurotransmitter acetylcholine. Now, the white line here represents the invaginations of the sarcolemma called the motor end plate. It's invaginated, so I know that this is the motor end plate of the sarcolemma because it's highly invaginated, means more surface area, more of the receptors that can bind acetylcholine, all right? So axon terminal. Another thing I want you to notice is that in the axon terminal, we see lots of mitochondria. In the motor end plate of the skeletal muscle cell, we see lots of mitochondria. We also see lots of mitochondria surrounding the myofibrils within the skeletal muscle cell. It's because a lot of these physiological processes require lots and lots of energy. So lots of mitochondria will be present. All right, so just recap, here's our axon terminal with all of those synaptic vesicles filled with acetylcholine. Now the space between the axon terminal and the white sarcolemma, the motor end plate specifically at this synapse, this would be the synaptic cleft. So the synapse is a functional connection between the axon terminal of the motor neuron and the motor end plate of the skeletal muscle cell. They don't actually physically touch, rather they are functionally connected. And it's that acetylcholine, that neurotransmitter, that when it's released, will diffuse across the synaptic cleft and bind to the receptors on the motor end plate. All right? Okay, so upon stimulation by a motor neuron, these synaptic vesicles get released, acetylcholine gets dumped into the synapse, will bind to receptors on the motor end plate and initiate an action potential on the sarcolemma of the skeletal muscle cell. All right? So now let's talk about some features of more features of the skeletal muscle cell. Now, let's start with these. If I look here and we're looking at like a cross section through a skeletal muscle cell, all of these guys that are pretty much filling the entire skeletal muscle cell, these are all myofibrils. So each one of these is a myofibril, and myofibrils are composed primarily of our myofilaments, which will be actin, the thin filaments, and myosin, the thick filaments, along with regulatory proteins like troponin and tropomyosin, which will be more important when you talk about muscle physiology in your lecture class. But for us, these are myofibrils, and they're composed primarily of thick filaments, which would be the myosin, and thin filaments, which would be the actin. So I'm going to turn it back this way. And we can see, so if we look at, this would be like a longitudinal section through a myofibril, we can see the filaments that are thin and kind of orangish in color, those would be actin. The thick filaments, which are kind of this pink color, and they're thicker, those would be myosin. And what this is showing you is a sarcomere, all right? Which the sarcomere are the functional structures of the myofibrils. And it's those so along the entire length of every single myofibril, we have sarcomeres lined up back to back, okay? And in the PowerPoint, I talked about the anatomy of a sarcomere and what you need to know from that. And some of those structures you could see here. So here, if this is our myosin, our thick filaments, then from here to here, where we see only thick, this would be our H zone. This line straight down the center, this would be our M line, which anchors the myosin in place. From here, where we see myosin stop, and we see some of the actin, from here to here, this would be our A band, which consists of overlapping thick and thin filaments. From here to here, where we only see thin actin, this would be our I bands. So you can see some of these structures even in this model, but I talked about the anatomy of the sarcomere on a cartoon diagram in the PowerPoint. This structure right here would represent the Z line. So up here, so this would be Z line. The next one up here, if it existed, would also be, so from Z line to Z line delineates a sarcomere. All right, so let's talk about some structures that surround each of these myofibrils. Here, this orange structure, this represents a T-tubule or a transverse tubule. You can call it either or. And it does a pretty good job of showing you what it is right here. You can see it's a tubule, kind of has a hole in the center. So this T-tubule is an invagination of the sarcolemma, which is the plasma membrane of the skeletal muscle cell. So the sarcolemma dips down and forms these T-tubules which surround, so see all these orange structures? These T-tubules completely surround the myofibrils. That's important because when the motor neuron signals, sends impulses, releases acetylcholine, acetylcholine will bind to receptors and initiate an action potential along the length of the sarcolemma. That action potential along the length of the sarcolemma will also go down these T-tubules because they're part of the sarcolemma, part of the plasma membrane. And when that electrical impulse, that action potential, arrives at the T-tubule, it signals to these white structures on both sides. These white structures that are sandwiching the T-tubules, these are called the terminal cisternae of the sarcoplasmic reticulum. And if you look, the sarcoplasmic reticulum, it's all this white stuff, is an extensive network of this, of the sarcoplasmic reticulum. It's an extensive network that surrounds these myofibrils. What is the sarcoplasmic reticulum? Okay, what is that? It's a specialized form of the smooth endoplasmic reticulum. It's an organelle that specializes in storing calcium. So these terminal cisternae store lots and lots of calcium. So when that action potential goes down the length of these T-tubules, it triggers these terminal cisternae to dump calcium into the cytoplasm of the skeletal muscle cell. And when these terminal cisternae of the sarcoplasmic reticulum dump calcium into the cytoplasm, that is when we can have skeletal muscle contraction. It's that release of calcium that ultimately leads to contraction. All right, so structures I just went over: T-tubule or transverse tubules. This is where the action potential will be sent down the length of these T-tubules. They're right next to the terminal cisternae of the sarcoplasmic reticulum, which stores calcium. That action potential triggers the release of calcium from these terminal cisternae. Together, these three structures, T-tubule sandwiched between two terminal cisternae, collectively these are called a triad. All right, what are some other structures that you should be able to identify? So I'm going to turn this again to where you're looking at like a cross-section through the skeletal muscle cell. Here, this is the nucleus of the skeletal muscle cell, and look, it's pushed to the periphery. We've seen that in our histology slides. that skeletal muscle cells, they're long cylindrical cells, and they're multinucleated because they're formed by the fusion of multiple cells early on in development. But what did we see in those histology slides is that the nuclei are pushed to the periphery. Why are they pushed to the periphery? It's because the cell is so full with these myofibrils. Because it's these myofibrils which will contract, overall shortening the skeletal muscle cells, therefore shortening the skeletal muscle organs. Another structure you should be able to identify is all of this connective tissue down here, which surrounds a skeletal muscle cell. What's the connective tissue investment that surrounds a single skeletal muscle cell? This is called the endomysium. Right? So the endomysium is directly attached to the sarcolemma, and it surrounds an individual skeletal muscle cell. All right, and that's it.